Hey guys, if you are watching this, it is because things did not go well over the weekend with my grandpa. He had a pretty rough go of it, and he has either passed or he's very, very close at this point. So just kind of bear with me here. If I go too quick as we go through, just tell the sub to pause. And as always, I will post the notes on Wednesday, the student version. So if you miss anything, make sure that you grab those. I already have printed out for you and ready to go your diagram packet. So make sure that the sub has passed those out. Follow along with any of those diagrams as I kind of explain them and go through them. And we'll just kind of make this work the best that we can. So um, chapters 8 and 9 are both on the nervous system. Chapter 8 focuses more on the peripheral nerves and just kind of the basics of what is a neuron and those kinds of things. So we'll be talking more about the overall nervous system, not necessarily the brain. Chapter 9 focuses more explicitly on brain anatomy, and you guys will do your sheep brain dissection and all of that with that chapter. So this is more, what's the difference between the autonomic nervous system and things that are involuntarily controlled versus things that are voluntarily controlled, etc. So that is our main focus this week. So when we look at functions of the nervous system, you guys will see several similarities as we go through, as I start to kind of explain the junctions and how our neurons work and everything. You'll see a lot of similarities between this and muscles, okay, especially when we were talking about the neuromuscular junction and how that works, and neurotransmitters are traveling between synaptic clefts and all of that. So hopefully this will make some sense to you guys when looking at the actual functions of the nervous system. So these top three bullet points should ring a bell. We've already talked about these in kind of other scenarios. So we have our sensory input, where our sensory receptors are going to respond to stimulus, right? So we've talked about a couple of times now the example of, putting your hands on a hot stove or whatever, and you didn't know that it was hot. Okay, you have those sensory receptors that are, in the skin. So that's going to send a signal to the muscle, eventually telling you, hey, move your hand, it's hot. So the sensory input is going to sense that. Then we have an integration neuron, and this is going to connect your sensory neuron to your motor neuron. So most of the time, these are going to be found in either the brain or the spinal cord. So think more kind of the central nervous system. This is where it's going to get the actual instruction to do whatever the motor response is going to be. Okay, so that motor response is move the hand. Okay, ow, it's hot, move your hand, respond to whatever that stimulus is. So those are three very incredibly important functions of the nervous system, and you'll kind of see that over and over throughout this chapter. Obviously, other functions then with the nervous system, mental capabilities, whether it is involuntary, our heart is still beating and we're breathing, and all of these things that our body's doing that we don't have to think about. Versus, you sitting here right now taking notes, voluntarily writing something down. So that takes a different type of mental capacity to do that. So we'll talk about that more when we get to the brain, chapter 9, but we'll kind of touch on that in this chapter as well. And then we've got homeostasis, right? We've drilled this over and over throughout the year. So with homeostasis, we're maintaining, all of the consistencies throughout the body. So making sure that our body temperature is staying the same, that our water levels are normal, that our pH levels are normal, oxygen, all of those different things are being balanced throughout the body. And the nervous system has a huge influence on kind of understanding where the body might be lacking, or where something's kind of an overdrive or whatever. So it's the nervous system's job to sense that and make the change. So when we talk about our nervous tissue, okay, so nervous tissue includes neurons, and neuroglia. Okay, glia is going to be more in the brain. So we're still going to talk about the different types of glial cells, but just kind of keep in mind that's going to be more with brain, whereas neurons are going to be more throughout the body. So our nervous tissue, obviously the main goal is to transmit a nerve impulse, all right? And like I said, think back to neuromuscular junction. So when we're transmitting a nerve impulse, I think you guys can see my cursor. So what you're going to see is the ends here, your axon terminals. At the very other end, if I had another picture of it connecting, your dendrites, which would be this part up here, would be right here. Okay, and they're not going to touch. You're still going to have that synaptic cleft. And that synaptic cleft is going to allow for all of those neurotransmitters to flow. So just like acetylcholine was flowing when we talked about the neuromuscular junction, in this case, we're going to be talking about serotonin, we're going to be talking about dopamine and any of the other types of neurotransmitters that, will be going throughout the body to send different thoughts and different actions and things like that. So that would be the third bullet point, to connect to other cells and eventually cause a response. So neuron characteristics, a neuron is a nerve cell. They require a ton of oxygen and they require a ton of glucose. Okay, they need a lot of energy. It makes sense that they need oxygen, right? If you've ever watched any medical show out there and somebody's been without oxygen for 30 seconds to up to 5 minutes, you're probably looking at least some deficiencies with the brain, if not completely brain dead and that kind of thing. So we need oxygen excessively with these types of cells. Like I kind of mentioned on the previous slide, the entire purpose of them is to receive and process whatever the input is and then produce a response. And that can actually happen on just a singular neuron level. So my signal is going to come in to my dendrites. Okay, the cell body is going to kind of process that and it's going to send the signal down my axon to my axon terminals, okay, the bottom part of my neuron here, and that's going to send the signal on to the next. So the dendrite receives and the axon is going to elicit the response. Okay, it's going to continue the signal on to the next cell. So it's going to continually transmit those impulses from neuron to neuron. So when we look at these structures, just to kind of break it down a little bit, so the dendrite, like I said, literally gets its name because it looks like tree branches, and dendro means trees. So this part here is where the stimulus is coming in, right? So there would be axons that would be kind of connecting with the dendrites here on all of the sides. Then you have the inside section here that's got the nucleus. So this is the cell body. It's also called the neurosoma, right? Soma means body and neuro means nerve, right? So this is the cell body of your neuron. So this is kind of where the magic happens. This is where it processes what's going on, right? And you can see, and you got a very large purple nucleus there for you to look at. Then you have the axon, okay? And it's this entire kind of tail that goes down to these little branches that come off. So the axon itself is one very large bundle of fibers, okay, and that splits off. So this is going to transmit a stimulus to a muscle, to an organ, to either another neuron, if it's, continuing the path, wherever it needs to go. And you'll notice these little purple things here, okay, on the side. This is myelin. And depending on where in the body your nerve is, it's going to have these little myelin sheaths that go over it. And the whole purpose of that is to actually speed up a reaction. And we'll talk about some of the diseases that are associated with not having myelin and what that can do to the body a little bit. Okay, guys, you have this diagram in your packet, and I just liked it because it's kind of all encompassing. So it tells you, up here that the dendrites are the receiving end, and right, you have your nucleus and the cell body that's kind of doing the interpreting, and then it's sending that signal down to the axon terminals where it will send it to the next neuron or wherever it needs to go. It also has all of the components of the axon. So there are these Schwann cells that have this myelin sheath on the outside. And you'll also notice that there are kind of portions of that axon where there is no myelin, and those are called the nodes of Ranvier. If you guys have heard of MS, right, multiple sclerosis, that is a disease where the myelin is damaged. So kind of that protective coating that surrounds the nerves, that starts to disintegrate. It's going to cause inflammation, and it's going to cause some problems, with memory and things like that. So the actual myelin sheath itself is a fatty protective wrapping around the axons, and it's a phenomenal insulator. And so because of that, it's going to increase our action potential, okay? So it's going to literally speed up how fast our signals are being transmitted, okay? So the more myelin we have, the faster a signal is going to conduct. So that's a good thing. Then we have those nodes of Ranvier, right? Those sections on the axon that don't have any myelin. That's where your actual action potential is going to develop, right? We have to have a signal. It's kind of the same concept as the neurotransmitters jumping through a synaptic cleft. Okay, there's always kind of this jumping going on within the nervous system. And then, like I said, MS is where the myelin sheath is kind of disintegrating, and it ends up attacking a very specific type of glial cell called an oligodendrocyte. And we'll kind of talk about those here in just a second. Okay. Looking at your cell here, so, right, you have your axon in the center, okay? You have the Schwann cell that is surrounding it. You can kind of see a nucleus here, that purple. And then we have this myelin sheath around the inside, that extra layer of insulation. And you can see, so here's this oligodendrocyte cell, okay? They're part of the reason why this is created. So in people with MS, this is starting to degrade, and so it will start to attack those oligodendrocytes, which are just going to even further degrade the myelin sheath on each one of those neurons. All right, quiz time. Just kind of mentally think through these. So which part of the neuron structure produces the stimulus? 5, 4, 3, 2, 1. Okay. Which one processes the stimulus? Should have been the cell body. Okay. All right. Question number 2, the structure that insulates the axon. 3, 2, 1. Okay, should be the myelin sheath. All right, looking at some of our neuroglia characteristics. Okay, so these are kind of the support cells for neurons, and there's different types. They're more numerous than neurons, okay, because they are what's kind of nurturing our neurons. They can divide, they can produce more cells. So when we talk about cells that we kind of think of in the brain and in the spinal cord, if they get damaged, right, they don't do mitosis anymore. So that's going to cause paralysis and things like that. That's not going to affect your glial cells. Okay, those will still, continue to produce more cells. One of the most common types of brain cancer is a glial blastoma. So cancer of the glial cells, and they're usually pretty aggressive and not the greatest outcomes as far as cancer is concerned. So we'll kind of go through each part of those. But if you just look at that basic neuron over here, right, it looks a little different under a micrograph image, but we have our dendrites coming out. They're obviously much larger when you look at a whole neuron. You got the axon, you got the cell body, and then all of these little cells, okay, are on the outside. All of this kind of pink matrix supporting tissue, those are the glial cells. So there are 6 different types of neuroglia. All right, we'll just kind of go through each one of these here. So we have the astrocytes. They kind of look star-shaped. That kind of helps you remember kind of think of like astrology and or astronomy, and kind of going with the stars and constellations and that kind of stuff. They are the most abundant type of neuroglial cell. And where you're going to see this the most is in something called the blood-brain barrier. And we'll talk about that when we do the brain, but basically it's a giant network of blood vessels that protects the brain, right? It needs a ton of oxygen. That oxygen has to get in through capillaries. And so it's going to act as a protective barrier between kind of everything else that might be floating around, around the brain and actually getting into the brain itself. The second type of cells that we have are ependymal cells. So these are going to create our cerebrospinal fluid, okay? When they do a spinal tap, that's the fluid that they are testing for. You're going to see that when we talk about the meninges of the brain, where that's located, there is, so that fluid, this CSF, is going to be all surrounding the brain, and it's going to go all down the spinal column. Then we have our microglia. So these are the cells that are going to kind of act as like a white blood cell lymphatic component. So it's going to help to remove any bacteria that might be there, any cellular debris. That's kind of their main job. Then we have the oligodendrocytes, just talked about those. So their whole job is to produce that myelin sheath. Then we have Schwann cells. Okay, these also produce myelin sheath, but the difference is that these are going to do the job in the peripheral nervous system. Okay, PNS is peripheral nervous system. The oligodendrocytes are going to make the myelin sheath in the central nervous system, which is the brain and the spinal cord. And the sixth type are the satellite cells. So these are going to surround the cell bodies of our peripheral neurons. Okay, it's going to keep those insulated. Okay, here's just kind of a look at each one of these. So here's the astrocyte, right? Kind of a star shape to it, kind of looking at that. They're really close to the capillaries. Some of them can even kind of surround and wrap their legs around the capillaries. Those are called foot processes. So you'll see those there. Okay, remember that makes up the blood-brain barrier, so you're gonna see a lot of that. Those oligodendrocytes, okay, here's that cell, here's the axons with the giant myelin sheath, so there is that. The Schwann cells, kind of the same thing, so 2 or 3 slides back when I showed you that axon, okay, that would have been a peripheral nervous system cell, okay, so we'll kind of do it like that versus, the oligodendrocyte here, you're going to see this very consistent myelin sheath, but remember that is in like the brain and the spinal cord. Those kind of microglial cells, right? They're micro, they're very small, but kind of those bacteria producing cells. And then the last one, we have these little ependymal cells, and they've got some cilia on the other end. And then we have the basement membrane down here. Okay, just kind of looking at this more all together. So here again is that capillary. So our blood supply, and here's that astrocyte, right? The neurons here are these giant kind of tan blobs with that myelin, right, sheath there, okay? See the oligodendrocytes that are creating the myelin all along those sheaths. We got these little microglia cells and the ependymal cells here along the edge, kind of forming a border. All right, so looking at types of neurons, right, we already kind of covered this at the very beginning, just simply talking about function, but there is actually a specific neuron that goes with each one of these. So our sensory neuron, like I said, carries the stimulus from our sensory receptor to the central nervous system, so either brain or spinal cord. Then we have our interneuron, okay, I think between, right? So that's going to kind of process and interpret our stimulus. And then we have our motor neuron that's going to carry the stimulus from the central nervous system to the effector. Okay, so you guys have now seen this probably 4 or 5 times, so this should make pretty good sense to you. So just looking at this kind of in pictorial version here, so we have our peripheral nervous system, right? So we have our touch receptors on this finger, and it is sending that signal to, let's just say it's the spinal cord. This is our interneuron, Okay, so it's kind of our association neuron. And then that's going to send another signal back out to a part of the peripheral nervous system. So whether that's a muscle, like this example, whether it's to an organ, whether it's to a gland to, produce insulin or glucagon or something like that. But you're going to have that kind of effect happen based on whatever the sensory neuron is sensing. So how do neurons communicate with other cells? So we know about our sodium-potassium pump. We've already seen an example of this with that neuromuscular junction. So our neurons are going to send messages to the next cell by using potassium and sodium, moving those against their concentration gradients, okay? So depending on what we're sending the signal to, another neuron or a muscle or whatever, the exchange of those ions is going to change the charge of those, right? And so what ends up happening is we create these electrical impulses. Physically, we've got electricity flowing throughout our body, and those are what we call action potentials, okay? So we've got this, kind of chemical change happening in the body. So we've got electrons that are being gained and lost depending on which, part we're looking at. And so that's going to create this kind of electrical impulse, which is going to allow this action potential to take place. So how this is going to look, right? Okay, it all comes back to peripheral nerves and the central nervous system. All right, so in this example, we've got somebody stepping on a nail. All right, so the stimulus is pain, right? He's got a pain receptor in his foot. He stepped on a nail. Ow, that hurts. So we have our sensory receptors in the in the dermis, and so there are dendrites. Okay, they're going to receive that signal. It's going to send a signal all the way down. This is an extreme example of how long this neuron is, but it's going to send that into the central nervous system. Okay, this is where we're going to have that sensory integration. This is where it's actually going to interpret that nail hurts. All right, so this is kind of that interneuron, right? So we have that sensory neuron here, the interneuron, and that's going to create that motor response. All right, so we have this interneuron here, okay, within the central nervous system, and the end of that axon is going to, connect here. We're going to have some synapses going on, and that's going to create a response in my muscle that's physically going to move. So the entire purpose of this is that these action potentials, this electrical impulse, is going to convey information. Okay, so an action potential. So these are electrical impulses of our neurons. So like I said, it's electricity, plain and simple. Travels about 1/40th of a second, which is really quite amazing if you think about it, right? Like, just think about all of the things. I mean, you guys are listening to me, you're seeing this with your eyes, you're writing, like all of these different things that you're doing all at one time, and how fast the nerve impulses travel for you to understand language and for you to see things and for you to hear. It's really just quite amazing. And then you have a response of writing all of that down, and just how quickly this actually happens is just so incredible. When we look at the electrical charges, we are measuring these in millivolts. Okay, so when you're looking at the little chart up here, okay, voltage, you have MV for millivolts, all right, and you have your kind of threshold sitting here. We have a stimulus, and that's going to there's two components to this called depolarization and repolarization. Then you kind of have this little resting time here before the next wave. This might look very similar if you just look at this right off the bat of an EKG. Right? If you're looking at somebody's heart, you have kind of the same wave pattern, and that makes sense because the heart is also controlled by electrical impulses from the nervous system. So obviously we'll talk much more about that when we get to cardiovascular. So usually this is done by chemicals that bind to the axon's cell membrane, and it requires that threshold, okay, kind of this baseline, that threshold has to be reached, okay? If the threshold isn't reached, this action potential is never going to take off. So here's kind of the stages of this. So before the action potential is created, okay, we're kind of at rest, right? Resting phase. And basically, right, if this repeated, you see resting state here, it would kind of just boop, and here we'd have multiple of these, just like I said, kind of like an EKG. So then step 2, we have the generation of the action potential. Okay, so that's kind of this big peak here. Then we have the propagation or the movement of the action potential. And then we have what we call repolarization. Okay, so this is afterwards, getting down to kind of our refractory period, sort of our kind of resting state. Okay, so we rest, we generate that potential, and kind of keep moving that forward, and then we're going to kind of repolarize back to resting, and the cycle will continue. Okay, so just kind of looking at this in picture form, there was a video that ICC had embedded into this, but I just don't really think it's going to transmit well into these videos, so I can just post it on Google Classroom and you guys can watch it. So just kind of explaining this to you in those 3 sort of basic steps. So step 1, we have this resting membrane potential, right? So what's going to happen is our sodium channels, the pink ones, are going to be present, okay? And a purple are the potassium. So those sodium channels and the potassium channels are going to start closed, okay? And you can see that up here, right? They're all kind of shut. They're not getting through. So the outside of the plasma membrane is going to be positively charged, okay? And the inside is going to be negatively charged, comparatively speaking, right? So when this depolarization happens, okay, so this kind of, the peak is now kind of starting, right? We're going up to the peak. So depolarization, we have those sodium channels that are starting to open, okay? And our potassium channels are starting to open. And so depolarization results because the inward movement of sodium makes the inside of the membrane positive, right? Okay, think back to diffusion, high to low concentration. So up here during resting stage, we have a ton of sodium chilling out here, and we have the opposite. We have a ton of potassium in here. So as soon as those open, those sodium are going to naturally diffuse into the cell. And then during repolarization, right, the opposite side down here, the opposite side of that peak, we have repolarization, which is where we have some of our sodium channels that are shutting, but the potassium ones are still open. So that's going to allow for our potassium to diffuse out of the cell, making sense, right? Getting that back to resting state. So this is back to a homeostasis kind of issue, right? We want to make sure that our ion levels are the same in and out of the cell. So the sodium movement into the cell will totally stop for repolarization, and potassium movement out of the cell is going to increase. Okay, so here's kind of what's going on in each of the action potential stages. And if you guys just want to, you know, kind of wait on this one to copy it down and just take a picture of it later on, that's totally fine. So when we look at the resting, okay, right, resting phase, there's no stimulus. And sodium has that higher concentration outside of the cell membrane, okay, so we have a positive charge. Potassium has a higher concentration inside of the cell where we have a more negative charge, okay? The charge is about negative 70 millivolts at this point in time. And if you watch that video, it'll kind of explain that in a little bit more depth. Then we have the generation of that action potential. So we have a stimulus present, whatever that is, okay? You flip on a light, there's a crash, you smell something, right? Any type of stimulus. At this point, remember we have to reach that threshold, okay? The threshold is minus 55 millivolts. Once we get to that that threshold of level, that action potential is going to take off. All right. That's going to start the depolarization process. So those sodium channels are going to open and sodium is going to flow into the cell. And as that happens, the cell is going to become more positive. Okay. Then we have this kind of propagation or moving phase, and this is occurring closer to the cell body of the axon. The action potential is going to move down the axon, right? We have this jumping kind of action now, and that's going to travel. It says a domino effect, but I almost kind of like to think of it as kind of jumping from one of those kind of myelin sheaths to the next, and it's only going to move in one direction. And then the last is the repolarization. Okay, so at this point, the stimulus is removed and those sodium channels are going to close. The millivolts is going to drop. We have the sodium-potassium pump that's restoring its balance. So it's going to pump out that excess sodium. It's going to pump in the excess potassium from the outside of the cell. And that's one of the active transport processes, right? It uses ATP. The sodium potassium pump was one of those that we learned about, back last semester, chapter 4. Okay, so just kind of a picture of how this is going on, right? So we have our extracellular fluid out here. We have our intercellular fluid going on in here. So we can see the difference in the concentration of sodium outside of the cell versus the concentration of sodium inside of the cell. Okay, sodium is the orange versus potassium, right? Potassium is blue. The concentration of potassium inside of the cell versus outside. So when we think about diffusion, even though we have our, ion channels, that's a type of passive diffusion. So we're always going to be thinking, that movement from high to low. So these steps in the action potential, and I want to give you guys plenty of time to get this written down, kind of word for word, just kind of like we did for muscles. So number 1, our membrane is at rest. Because we have that positive charge outside, we have a negative charge inside, okay? So this is where we have no stimulus. Step 2, we have a stimulus, and it's going to trigger the opening of the first few ion channels in the cell membrane. And that's going to allow for sodium to enter into the cell. If the stimulus is strong enough, that's going to send an action potential. Okay, so I'm going to use kind of a gross example of this. So you poop, right? It smells. So you don't smell it right away. It takes a few seconds, and that's not necessarily because those smelly methane molecules aren't there. It's because the quantity of them isn't strong enough that your nose is going to sense them. As soon as your nose senses them, right, it gets that stimulus, and boom, we have an action potential telling you that, ew, this smells totally disgusting. Same thing with less gross things like when you're baking cookies or something like that. But just kind of think about it as that you have to have a strong enough stimulus for that action potential to ever be generated. We have to reach that threshold. Step number 3. So as that happens, right now we've entered this threshold has been reached. So now more ion channels are going to open. As more ions open, channels open, we have more of the sodium that's going to enter the cell. And so now we're looking at the differences in the charges, right? So now the inside of the cell is becoming more, more charged, and that's going to set off depolarization. And then step number 4, we have this electrical change going on. So this is going to kind of trigger the closing of the first ion channels and the opening of the second. Okay, so the sodium channels are going to close, but those potassium ion channels are going to open. Okay, so this process is going to continue until that stimulus is removed. Okay, you guys have this in your notes as well. I like it pretty much, again, step by step, and it explains, so step one correlates with one on the graph. So number one, we have the stimulation of the neuron. Two, here's that threshold. So we get enough of a stimulus that it's actually going to cause an action potential. Three, we have this influx of sodium. Four, we have the membrane voltage is going to read about 35 millivolts. By the time, at this point, all of the sodium channels are closing. And at the same time, most of our potassium channels should be open. Okay. 5, potassium is going to leave. And then 6, we're kind of at rest. Okay. So looking at the conduction of action potentials in a myelinated versus an unmyelinated axon. Okay, so these nerve impulses are obviously extremely rapid, right? We know that they, kind of hop from one part of the nerve to the next. And myelin's going to speed that process up. But when we have an unmyelinated fiber, it's still going to conduct a nerve impulse, but it's not going to kind of hop how we might think of. It's going to be more of a kind of solid fluid impulse down the entire length of that nerve fiber. Myelinated fibers, right? So here's that. Okay, we've got our little nodes of Ranvier, right? So they're kind of hop and hop and hop. Well, if we don't have this myelin on the axon here, right? It's just going to be one smooth kind of transmission from the soma to the axon terminals. Okay. The saltatory conduction is the jumping of nerve impulses. And so when we have our myelinated fibers, this happens, quick, between 3 and 15, meters per second, versus in unmyelinated, okay, for 2 meters per second. All right, so way, way faster than that. So we obviously want those myelin. Like I said, if you think about people with MS, they're not going to have as much myelin, which means that their nerve impulses are going to be much slower. So they're going to end up, having conditions where, maybe they have some tremors, or they kind of seize up a little bit, or they have memory lapses and things like that. Okay, this image is basically what that video is just kind of showing you. So we have our membrane that's ready to be excited. Okay, so this would be at rest in the green. And so we have that action potential in progress, right? So that signal is being sent from dendrites down to the axon. So, and you can kind of see this is a step-by-step progression of that happening. So it starts here, it moves a little bit further, moves a little bit further. But following that action potential, we have the refractory period, okay, when that potassium was coming back and kind of restoring balance. So that's always going to follow that action potential until eventually we get back to our resting state. Transmitting our nerve impulses to the next cell. So we have a synapse where our nerve cells are going to meet each other. So our neuromuscular junction is an example of that. We have presynaptic neurons and we have postsynaptic neurons. Presynaptic neurons are going to, carry an initial impulse. So they contain synaptic vesicles, which have our neurotransmitters in them. And then we also have our postsynaptic neuron, and that's going to be the next one to be stimulated. Okay, so if our presynaptic neuron is, right here, here's our dendrite axon. Okay, here's the next dendrite. So that next dendrite would be the postsynaptic neuron. So obviously there's always going to be a progression of presynaptic neuron, the synapse actually happening, and that neurotransmitter being transmitted to the next neuron, which is the postsynaptic neuron.